Hello and welcome to our next topic of conversation here in microbiology, and this is going to focus on Chapter 8. The title for Chapter 8 is Microbial Genetics and Genetic Engineering. And so genetics is a topic I find that quite a few of our students here at Calhoun, I know everybody's coming from diverse backgrounds in terms of ages and when you were in high school and so forth, and exposure to various science topics in the past. But I find that a lot of our Calhoun students don't have a very good background in genetics. And so I am going to approach this chapter from the standpoint of assuming that you've had little or no prior exposure to this topic. Now, genetics, as you guys know, is a hot topic for the 21st century. I know probably not very many of you are planning on working directly in the field of genetics, but we're undergoing a big shift in medicine and diagnostics in this country, where a lot of these things are going to be moving in the direction of applying genetic technology. So you guys who are going into nursing, for example, not too far down the road, you're going to be giving your patients DNA-based medications or medications that use engineered viruses to deliver products to very specific parts of the body or maybe cancer cells to try to kill them. So because of that, it's really important that you have some background information on this subject, because if you're planning on going into nursing, you're going to be the one who's bridging the conversation between the physician and the patient. So you need to have some background in this subject area. And on top of that, a more immediate concern is many of you have to take entrance exams for health sciences programs, and I'm especially thinking of the TEAS test for nursing, and there are questions on this subject on the TEAS test, so you will get some of the background information for that as part of this course. All right. So now several of these, I'm trying to link things to the sections in your textbook so you can tie together the reading with these video lectures. However, some of these Chapter 8 sections are pretty long and involved, especially if you're coming in with little to no background on this topic. So I'm going to have to chop each section of the text into multiple video lectures. All right, so just to kind of get started here, just some definitions. This term genetics, what does that mean? It's the study of inheritance and heritable traits that you can inherit or transmitted from parent to offspring. And we've already talked about before how DNA is the molecule of genetics, and it's the inheritable molecules. Those are the genetic blueprints that have to be passed down from generation to generation. And that's true across all living things. All life forms that we have discovered on Earth use DNA as their genetic material, and that's the location of their genes, or these sets of genetic blueprints. All right, again, so when we hear genetic material, that's these sets of genetic recipes, codes that are ultimately going to be responsible for the traits of that organism. And a trait is any kind of physical characteristic you can observe about an organism. So think back to when you were doing the unknown bacterial identification lab activity. You ran all those tests on unknown bacteria, and any one of those things that you could observe was an example trait. Or the gram stain, the morphology, the colony morphology of bacterial species when they grow on agar plates, temperature preferences, pH preferences, oxygen requirements, all of those things are traits, and you can trace these traits back to the DNA molecules of that organism. They really do function, as we'll see, like a set of chemical blueprints that are going to be responsible for the traits in living things. All right, so interestingly, much of what we know today, now across all living things on the planet, the viruses have exceptions. Remember, viruses, we don't categorize those as truly living things. They don't meet all the characteristics of life. But all cellular life that we have discovered on Earth, the basic core way that this function of genes on DNA is blueprints, and that leading to traits in that organism, it works the same across all cellular life that we have discovered so far. And much of what we understand about these genetic processes has come from studying microbial genetics. And that just kind of makes sense because microbes are a whole lot easier to work with in a laboratory. If you want to run genetic studies on bacteria or yeast, you can grow those up pretty quickly and fairly inexpensively in a laboratory. So much of the basic information that we understand about genetics today was originally worked out in microorganisms. All right, some other terminology here. This word genes. Genes are specific locations on DNA molecules that are serving as a blueprint or a code. And just to kind of emphasize that. Now, what kind of cell is this? Prokaryotic or eukaryotic? And hopefully you're thinking eukaryotic because you see a nucleus there. And you've probably seen this before. This is the typical drawing of a chromosome that you see in any kind of biology or life sciences class. And a chromosome is really a DNA molecule complexed with proteins that help condense that DNA down and wrap it up and keep it all nice and folded so it can stay within the confines of a cell. And as we'll see, DNA molecules are huge. If you actually unravel them and took them out of a cell, they are far, far, far longer than an individual cell. So DNA has to be all wrapped up and condensed down so that it can fit inside a cell. And so when we're talking about a chromosome, we're really talking about a DNA molecule complexed with proteins and all wrapped up around it to help keep it inside the cell. Now, when you guys see this type of picture, this is actually a duplicated chromosome. So if you've studied mitosis before, which is the process of copying chromosomes in a eukaryotic organism and making sure that individual copies, one copy of each, goes to the two daughter cells that will form when the cell divides. That's mitosis. And so this is a duplicated chromosome that's undergoing the mitosis process. So those two copies are actually going to separate from each other when a cell divides. And a bacterial chromosome is not going to look like that. So just keep in mind there are differences between eukaryotes and prokaryotes. But what we're seeing here is, when you see this, and you could actually see those in a typical science laboratory looking through a light microscope, because when DNA gets very, very, very tightly wound up and compacted down and in this duplicated form, that occurs, when a cell is undergoing mitosis, and the DNA is so wadded up and folded up and condensed down, you can actually see that with a light microscope in a laboratory. Otherwise, you can't see DNA with a typical light microscope that goes up to 1,000x total magnification. You would need an electron microscope to be able to see DNA otherwise. All right, what they're trying to show you here is, if you kept pulling this out, pulling this out, pulling this out, pulling this out, eventually you would get to that double helix, double-stranded DNA molecule shape that's so familiar. But this is much, much, much more zoomed in than this. To actually see a double helix structure like that, you would need, an atomic force microscope, which is the most powerful type of microscope out there. So some other terminology. Now, genes are located on DNA molecules. So if this is a long DNA molecule, double helix, double-stranded, there's a stretch of A's and G's and C's and T's on that long DNA molecule that's going to function as a gene. It's going to be a blueprint. And typically when we're talking about basic genetics, we're thinking about these genes as being blueprints that are going to tell the cell how to make a particular protein. All right, another term, genome. This is a more contemporary term. All of the genetic material of an organism is called its genome. So maybe you've heard of the Human Genome Project. So that was a project about 15 years ago where the AGCT letter code of the entire human genome, all of the DNA in an example human cell, was determined. Billions and billions and billions of base pairs of DNA. And so the human genome, your genome, is all of the DNA present inside your cells, all the genetic material. All right, and as you guys have probably heard before, the human genome is diploid. That means we have two copies of each of our chromosomes. And remember, we have chromosomes 1 through 22, two copies of each of those. And then you've got your sex chromosomes, 2 X's if you're a female, XY if you're a male. And then we have, in bacteria, as we've talked a little bit about before in this class, they typically just have one chromosome, one large DNA molecule, and it's usually circular, which means the ends of the molecule are connected. Our DNA is linear, so it's not closed together as a circle. Some other terminology, genotype. Maybe you've heard genotype and phenotype before. This term phenotype refers to traits. Genotype specifically refers to the genetic information, these chemical blueprints that exist on the DNA. And as part of genetics, we say genotype encodes phenotype. So genes serve as a code, kind of like a chemical letter code that instructs the cell how to make proteins. And proteins, we've talked about those before. Those are the chemical workhorses of cells. They're very diverse. They have lots of different functions. Many of them are enzymes. And ultimately, these proteins in cells lead to particular phenotypes or traits. And we'll see lots of examples of those as we move forward. And an example that we could talk about right away, if you're a bacterial organism and you're capable of doing fermentation starting with lactose, you've got to have the enzymes that allow you to do that, to allow you to undergo that fermentation process. Enzymes are proteins. So if you are a bacterial cell that makes those enzymes, you've got to be able to trace it back to your DNA. You've got to have genes on your DNA that provide the chemical blueprints that are going to direct the production of those enzymes that allow you to ferment lactose. I know some of this is kind of abstract at first to wrap your mind around, but it'll make more sense as we move through. So this term genome, this is a figure from your textbook, which is pretty good. It's just showing you that genomes can have different characteristics and in different cell types, and then also in viruses. Viruses, even though we say they're not truly living things and they're not cellular, they do have genomes. Their genomes contain the genetic blueprints that are going to provide instructions for making the parts of the new viruses. And we're going to get into viruses in a lot more detail in Chapter 5. All right, so here's a generic eukaryotic cell. And notice your textbook is calling this a composite cell. So this is not a cell that really exists because it has features of different kinds of eukaryotes shown all here together. But a genome in a eukaryotic cell, depending on the type. Now, all eukaryotic cells are going to have chromosomes and a nucleus. And those are going to be where the bulk of the genes are located. These genetic blueprints are going to be on the DNA chromosomes and the nucleus. Now, some eukaryotes have little extra pieces of circular DNA. Those are called plasmids. You may remember we talked about those in Chapter 3 when we were discussing DNA of bacteria. Chloroplasts, those are organelles, little membrane-covered compartments that exist in eukaryotic cells that do photosynthesis. And chloroplasts can have some of their own DNA with genes on them. The mitochondria that exist in eukaryotic cells actually have their own genomes that have genetic blueprints on them that are responsible for some of the traits of mitochondria. You may also remember that we've talked before about how the theory of endosymbiosis teaches us that today's mitochondria that we have in eukaryotic cells probably evolved from some sort of symbiotic relationship that was set up between a bacterial cell and another type of cell in the distant past, which was probably an archaean. Okay, in a prokaryotic cells, like bacteria, which we've talked about before, bacteria typically have one circular chromosome, but then they may also have additional little small circular pieces of DNA that are called plasmids, and we talked a little bit about that in Chapter 3. Viruses tend to break some rules in genetics, so some viruses have DNA genomes, but some have RNA. So viruses break some of the rules that we talk about. And by rule over here, I mean going from genes on, if you're going to make a protein that's going to lead to some aspect of the phenotype or the traits of an organism, there's a flow of genetic information where you've got a gene on DNA that gets copied onto an RNA molecule, that gets read in the cell to make the protein, and the protein leads to the phenotype or the trait that's associated with this protein. So you've got a flow of genetic information that we're going to be discussing. And viruses sometimes violate that flow a little bit because there are viruses called RNA viruses, and all their genetic information, their genetic blueprints are located on RNA molecules rather than DNA. But cells, again, all cellular life forms that we know about have DNA genomes. All right, this is just reviewing, again, some information from Chapter 3 in a prokaryotic cell, and when we're talking about prokaryotes in this class, we're generally focused on bacteria. You've got that one larger DNA chromosome, and some bacteria have plasmids, these little extra pieces of DNA. And the chromosomes are going to have the crucial, critical, mandatory genes that are necessary for life. Plasmids will have generally extra genes that give that bacterial cell some sort of a survival advantage. So we're talking about genes as being locations on DNA that code for proteins. A typical bacterial chromosome may have hundreds to thousands of genes that code for different proteins. Plasmids will have far fewer. Usually they'll just have a few genes that give that bacterial cell some sort of a survival advantage if they exist. All right, so, I just remembered something here. I noticed under plasmids here, a few thousand to a few million BP. BP stands for base pairs. So if you're thinking about a DNA molecule, A on one side paired with a T over here, and then here's a G and here's a C, here's a T and here's an A. Each of those is a base pair. So the lengths of DNA molecules are usually described in base pairs, how many base pairs are present. So plasmids are typically a few thousand to a few million base pairs in length. Now when you get to bacterial chromosomes, you're going to be up in the millions somewhere. And when you get to human chromosomes in our cells, you're getting into the tens of millions to hundreds of millions of base pairs per DNA molecule. All right, so again, this flow of genetic information, this is going to be something that we talk about in Chapter 8 and Chapter 5 as well a lot. As we're going to see, how do we go from DNA to RNA to protein? What does all of this mean? I mean, DNA is an organic molecule. How can that serve as a blueprint? But it really does, because you've got these sets of these chemicals, adenine, guanine, cytosine, and thymine. Those are capable of functioning almost like chemical letters, and there is a letter code that is going to direct the cell to take this information in DNA, and first it gets copied onto RNA molecules, which are also nucleic acids, and then the cell reads these, and by reading that RNA, it knows what amino acids to link together to build a protein. So we're going to be changing languages as we talk about this, from the nucleotide language of DNA and RNA to the amino acid language of protein. We're going to see there is a genetic code that has been worked out that exists across all cellular life forms that functions as a set of rules and decoding for how you go from this DNA language here to the protein language over here. Again, phenotypes, we've talked about that. A phenotype is any kind of observable trait. So just think about some of your own phenotypes that you have. You might be somebody who has a real high metabolism and burns up calories really easily. Lucky you if you are. That's an example phenotype. Your skin tone, your hair color. Not that we care much about hair. Hair is not important. Your eye color. Are you more likely to develop heart disease as you get older? All of those types of things are phenotypes that could be traced back to your DNA, genes, genetic blueprints on your on your DNA. And I already mentioned some phenotypes or traits that we have observed in your virtual lab activities that you've undergone. I do want to point out here there is a link to a set of basic genetics tutorials from the University of Utah. I'm not going to run these on a YouTube video lecture, but I encourage you to click on that link in the PowerPoint and go there, because they have a set of very basic tutorials that are really good for, in an interactive, animated way, kind of hammering home some of these introductory concepts associated with genetics. So I encourage you to go ahead and take a look at that. All right, so that was kind of a brief introduction to get us started with genetics, and in the next video lecture we'll continue this story, and then soon we're going to be transitioning over to, how do we go from this DNA language to this protein language that leads to the traits that living things have.